Good evening, C.R.E.A.T.E community, and good evening to everyone who's new to C.R.E.A.T.E webinars. Welcome to all of you, and thank you for sparing 1 hour to be with us this evening. Today is our eighth C.R.E.A.T.E webinar. We started the whole journey last September, and I'm very happy and pleased to continue this journey with all of you. Our group, our first or initial group of webinars aim to establish kind of shared understanding of the core concepts in research and to raise the awareness about the importance of discussing and continuous education in research. As we move further, the second group of webinars is more about selecting specific and interesting topics that we discuss in depth. And that's the reason for our meeting today. I think for the people who are attending the webinars, they know who I am. I'm Bashar Mohammed Musa, the program director. I'm associate professor at College of Medicine, deputy director of US Diabetes Center. I'm coordinator of the master program in diabetes management at University of C.R.E.A.T.E as well. And I'm very proud to always talk about this initiative as a very successful learning initiative between University of C.R.E.A.T.E and Emirati Diabetes Endocrine Society. So C.R.E.A.T.E is about thinking, sharing, discussing all aspects of research. Just few instructions to make sure, as usual, to make sure that our webinar is moving smoothly. So the webinar is live streamed now, and it's available in the EDES YouTube channel. At the end of the webinar, we will have Q&A session. So please make sure that if you have any comment, question, or feedback, please just make sure that you write it in the A and Q box. On-demand webinar will be available on the EDES Institute after the webinar. It gives me actually a great pleasure, and I'm very grateful to Prof. Mark for being here with us today. I know he's a very busy person, but he spent this time to be with us in C.R.E.A.T.E webinar. Prof. Falaji, he is a professor and chairman of the Department of Pharmaceuticals and Pharmaceutical Technology at the College of Medicine. He is a founding leader of the Drug Discovery and Drug Delivery Research Group at University of C.R.E.A.T.E. And also he's founding director of the Center for Drug Discovery and Development at Nova Southeastern University, Florida, USA. He also received his PhD from University of Manitoba in Canada and joined QLT Pharma in Vancouver, Canada, as a Natural Science and Engineering Research Council Fellow. He's have amazing kind of journey when it comes to development of a novel sublingual ODT platform as an alternative to the invasive delivery of several drugs. He filed more than 84 patents as the main inventor at various US, Europe, and Asian patent offices, of which 24 have already been issued. He has three licensing contracts with the pharma in US, Europe, and his inventions, and several other research contractors. He published more than 64 publications, and he has about five book chapters. Um, he's doing a great job, I think, and he's a great leader. He works for different entities, including, like Ministry of Health. He's been awarded different prizes, including Mohab Innovation Award, US Research Incentive Award, and Allergy Research Award from the American Academy of Allergy, Asthma and Immunology. He was featured in several local, international TV channels, media, and websites and magazines, including CNBC, Newsweek, Life Science, and Popular Sciences. His main interest when it comes to research, including formulation and development of novel dosage platforms and the application of nanotechnology during in drug delivery through non-invasive routes. An amazing kind of contribution to research, and we are so pleased to have Prof. Mark today to talk to us about the discovery from the scratch to the approval, understanding the stages of translation research. Thank you very much, Prof. Mark, for being with us today, and the floor is yours. I think you are muted, Prof. Mark.
Okay, I think I'm back now. Okay, so
Yes, you are.
I was saying, thank you very much for the invitation. Thank you, Professor Pashai, for this beautiful introduction. Uh, it's my pleasure to be part of this webinar, part of the C.R.E.A.T.E. It's a very wonderful series, and I'm very grateful for EDES also for organizing the webinar. Today, basically, I will try to share some of my previous experience and my kind of like, understanding for translational research. And I'll try to move, we have kind of like an extensive topics to discuss today. Hopefully they are helpful. And I just want to kind of like, share with you that these are not AI generated. So if you see any mistakes or any typos, this is a good sign, not a bad sign, that it's been shared or basically it's been man-made and not AI made. So just for today, we're going to cover a series of topics. So we're going to a little bit introduction about translational research, and also we get the chance to compare it or basically to discuss how they are different or similar to the product development. And then we'll talk about the stages of both translation research and product development. Also, we're gonna try to understand what kind of like the steps or some of the concepts or ideas that to establish successful translational research, and also how we can successfully execute these type of research and experiments, and how we can develop our project. So basically can lead to further product development, whether through licensing or startup. We're going to try to briefly understand the differences. Then ultimately we want to go to kind of like, the filing, the IND filing, trying to go through the regulatory pathways and what are the different components, including, of course, clinical trials, where we're going to talk about relatively like, some.
Sorry, Prof. Mark. I think we
Yeah.
don't see the slides
Oh, you
yet.
don't see? Okay.
Your slides is not shared yet, or maybe you shared it and maybe there is a technical glitch.
I'll try again.
Yes, please. Yeah, now we can see your laptop or
Okay,
your computer.
perfect. Okay. I'm
Yeah,
sorry. I'm
here
sorry
we go.
about that. So
That's all right.
that exactly what I was talking about. So these are the kind of like, the content for my talk today. And we were saying that we're going to go through some regularity steps that we need to go through for IND filing and then NDA. And I'm going to try to make a very, very short conclusion. So just to start, basically, as you know, that scientific discoveries kind of like take a very long time to kind of like transform this basic science into an actual practice, whether in a form of guidelines or whether in a form of actual product. For example, in case of product development, we can take up to 10 to 20 years, depends on the stage that you're starting from, and can take up to $2 billion in order to kind of like develop product. Same thing applies for if you want to try to change or update some policies and guidelines when it comes to the practice. It takes really a long time, 10 to 25 years around that time. So we see that there is a really significant delays in transforming these basic science into actual application. And you see that there is a gap between what we call the scientists, whether they are in academia or in practice, and the actual policymakers or basically the pharma industry that actually make things in the market. And the reason because there's two different brains, and everyone have their own sets of goals and way of approaching the research as well. They are using different languages, different terminologies, and time frame is different. For example, people in academia, they have luxury of time, but people in industry, they might want to kind of like expedite the process. So basically, we're working in two different environments, and we need to bring these two brains together, or we need to do something to expedite the actual translation of that basic science into actual application. In addition, of course, to the things related to the logistics and drug development processes, long processes, regulatory things, this adds up to the actual time. So where is actually the gap? We found that basic scientists is actually usually they're more interested in trying to understand and generate a general knowledge that can be useful, can enhance the level of understanding and learning about how things are getting done. But most of the time, you'll find that this type of research does not really answer the questions or basically lead to actual development product, cannot per se alone can address all the questions required to change a policy or to come up with the product or to approve a product. Yes, eventually, maybe after a very long time, you'll find that you can accumulate the knowledge that might lead to change certain guidelines, treatment guidelines, or product development. But usually that time is a very, very long time. So we need to come up with a way in order to kind of like, bridge between the basic science that's essential and actual application of that science. So they came up with the terms translational research, where sometimes you can see it known as bench to bench or lab to clinic. And in our case here, I called it discovery to approval because I'm going to talk about also some of the approval steps. So usually, like we'll see that in this situation, the research is actually designed purposely or designed to address specific issues, and basically from the industrial perspective or from a policymaker perspective or from practitioner perspective rather than from scientist perspectives. So they aim to accelerate and to address the questions that would lead you to conclusion and outcome. This outcome can be transformed into a product or transformed into new guidelines or transformed into change into in the practice. So basically, now we're conducting the research, but we are more focused or have better attention to what kind of questions that are expected from regulatory or from policymakers actually to be able to use that science and apply it. So you see here in this case is actually the translational research bridges between the basic science and the actual application and the clinical practice. So basically now we're doing more relevant type of research and activity, not to underestimate the general knowledge gaining from the basic science, but this is it's kind of like another type of research to serve certain purposes, which is to serve the community. It's basically based on the community needs for changing policies or treatment guidelines or even new product for unmet needs. So they are designed carefully. They are more organized. They're utilizing the financial resources and the manpower to kind of like have more something has a value or economical value or basically can convert it to help the general public increase the quality of life or even disease management or even come with a new product, which is going to affect the whole economy of the region as well. So this is why you start seeing that NIH, for example, the National Institute of Health they already started a center called National Center for Advanced Translational Research, or translational sciences aims to help expediting the process and bridging the gap. You'll see that a lot of institutions, for example, here I gave one example, Translational Research Institution in Arizona, for the same purpose. There's many examples. This is one of them. You see journals names translational research. So basically now this type of research is actually becomes more popular and can actually impact the public health faster, and actually can also contribute to the actual general economy. So what is the difference? If we want to differentiate, because we hear translational research and or research translation, and we see commercialization or product development. In case of commercialization, or in case of product development, which is my area of expertise, is basically it's one type of the translation research. So translation research is more wider, okay, can include multiple things. But for example, in case of product development, we are more concerned about outcomes that actually intended to generate some product that has a certain value, or even sometimes it's a service, it's a device, but basically something that tangible can actually impact patient health. It can generate financial gain. Most of the time, it's basically for profit in a form of licensing to a third party or even creating a startup. However, on the other hand, we find that translational research is wider. It's actually aimed to convert the basic science to a real world. That science could be in a form of like a new policy, in a form of new treatment guidelines that actually transfer to be practiced into the clinic, and then maybe can be done at a larger scale, kind of like, studies to see how it impacts the public health. Also part of it, commercialization, product development that I gave an example for. So what are the different stages for that research? So if we talk about translational research, usually they're classified into 3 to 4 stages. And the first stage is basically to bring that basic science and convert it into clinical trials. So moving that basic science to reach clinical trials. And now, and T2 or stage 2, they basically moving these clinical trials or the information from clinical trials to kind of like form some sort of like guidelines and basically new protocols, right? And then we move to the T3, which is now, it's basically you have apply these guidelines into actual practice. So move it into the clinic. So practitioner now they follow new guidelines based on that basic research that are actually translational. And the last stage is basically after you actually introduce it into the clinic and you have a lot of data coming from using this new guidelines. Let's say, okay, now you can make a broader type of assessment, like, usually they basically will know for health outcomes and effectiveness and so forth. However, when we talk about product development, I mean, we have slightly different stages. So usually we start with the discovery and development, and here usually we are needing pharmacologists, medicinal chemists, and the whole idea is that to identify a certain target, and then the medicinal chemist, they can actually optimize or find the lead molecules and they optimize them. At this stage, you can start filing patent. Again, here the purpose is commercialization, so we want to protect it. We're going to talk a little bit about patent filing later on. So basically, at this stage, because it doesn't have a real application or translation, so it doesn't have a lot of commercial value, but still you have to file some sort of like patent to protect the idea. Okay, and then you move to the next stage, which is preclinical studies. And here also still both consider our basic research, but we are more concerned with the characterization of these molecules, put them into actual formulation, and try the first step of translation, which is drug delivery. Try to do in vitro, try animal studies, and this is where you start building some value to that product. So definitely patent filing is important if you intend to commercialize, to protect the IP right. So basically, this is where you start to build value for these products. You can even maximize that value by doing some pilot clinical studies, although right now is getting more stringent to do clinical studies. Uh, but then ultimately, the next step is that you have to go through the regulatory pathway. So you need to file an IND, okay, and then you move to the regulatory stages where you actually file the NDA, where you are seeking an approval to market the product after you finish all the IND filing, including phase 1, 2, and 3. And then you can go to what we call, and all of this, like, I mean, the clinical trials is considered kind of like T1, T2, if you want to compare it to translational research. And then later on, after the approval, we get the T3 and T4, if you want to compare it to translational research, which is the phase 4 studies and pharmacovigilance, where you get more data, more information. You can do a larger type of assessment on larger population. So this is just the steps that I was talking about, that moving from discovery all the way to that post-marketing. And we're going to elaborate a little bit more about each part, especially the clinical trials. So how can we establish translational research project? So let's say I want to move on from the basic research that actually to gain knowledge, which again, I'm not underestimating, this is a very valuable kind of like, service to the public or to the field. But if you want to be more into the translational research, so first of all, you want to, as most of you know, that you need to find or identify a research gap. The best way to do it is basically you create area of specialization. You'll see scientists are working in different fields, different area, different diseases. That does not create an area of specialization. You need to be more specialized in order to be able to distinguish which is a real gap that can be actually have a value. Also, building a network of practitioners. The best ideas can come from the people who are in practice. What kind of like issues that they face daily about the application, whether it's a treatment, whether it's a protocol, whether it's a procedure, whether it's a device. So basically, you can get feedback from them all the time. So you having this practitioner network in your area of specialization is extremely valuable. This is something helped me a lot and during the process of my patent filing and product development steps. Then one of the ways also, although like, kind like, not really the best part is to go, if you're totally not aware where you're going to go, is to go through the review articles. So review articles, usually towards the end, you have something called limitations or future directions. Usually these review articles are written by people of high specialization. They're expert in the field and they can distinguish where are the future direction, and this can be the lead for you. Then after you have this research gap, you want to start to do more practical application, right, for that research. So basically, you want to identify where is the unmet needs, whether it's a treatment dosage form, or try to apply it in a form of improving a treatment, so you have a better drug profile when it's from the structure-wise and efficacy. You can try alternative routes that can be more convenient for patient. For example, you can have additional dosage forms that can like something that like sustain release or for example, targeted drug delivery. So these are more specialized areas, or you can improve a method like or a device. Although like usually in commercialization, product development as a drug is usually more attractive than a device. This is something I learned through my my research experience, where I developed some small device, kind of like to do a test that for something that with unmet what we call unmet needs. And then later on, we want to file patents. I would advise you're not going to find a lot of market for it. Interest is very low. Commercialization and marketing is very difficult for devices, so we stayed away from it. Then later you can create your research team. Now, after you know where's the gap, where's the area of specialization, what kind of things that you want to develop, then you start to gather your selection of your research team. And you want to kind of like, select people based on their scientific contribution and the infrastructure. what kind of facility they have, what kind of experiments they can do for you. You can select different people from different fields. It depends again on what is that you're trying to do. And consultants. So sometimes they are not a co-investigators, but they can be a consultant, especially practitioners or regulatory people. Or if you are like, for example, practitioner from a formulation person that can at least before you start give you a hint about what is the possibility of applying your idea or basically your concept. Okay, so this is one of the very quick, ideas about how you can start a translation research project. And then comes to the execution is very, very critical here, where you actually, that research, you need to develop a roadmap for it. For you need a project manager, usually in academia, it's the BI is the project manager, and you divide and delegate the work. It's much faster than rather than doing every step by yourself, especially if you require different expertise. You develop a research proposal. Once you develop that research proposal, you can run it by your consultant to see, especially regulatory, see actually if it's a product, then you're going to go through a regulatory pathway. So is this something feasible or something that's doable? It's easy or it's difficult? Of course, regulatory always difficult, but I mean, is it to kind of like, show you or guide you what would be the proper way to move with it. And then seek funds, which is sometimes is very difficult, and not all institutions understand the differences. When you're seeking funds, you are actually releasing your idea to the reviewers and maybe to certain level of the publics. That's not what you're supposed to do in translational research, because you're supposed to patent the idea. Okay, later on, once it's been reached a certain maturation or mature to the level where you can actually file a patent. So here, yes, you want to get funds, but at the same time, you want to be very careful. If there's no confidentiality agreement by the reviewers through the institution that actually provide the funds, maybe this is something that you need to be careful with. And then you start executing the experiment, and here you're doing the basic research. I mean, if we're talking about researchers in academia, then actually we're doing mainly basic research. However, this basic research can be focused to serve later on, in the regulatory pathway or basically in the product development. So you want to develop these studies to meet industrial expectation, to meet pharma expectation, to meet regulatory expectations. So basically, you can do tons of studies, but you want to do them purposely to meet their understanding or basically what they're looking for. Imagine what's the industry would ask you or the pharma would ask you in order to kind of like license the product from you. Then you need to make sure that you're addressing these questions. You need to perform studies from the get-go according to the pharmacopoeias standards, because these are the standards that's actually from the regulatory perspective is acceptable, or according to the ICH guidelines. For example, if you have the ability to do JLB type of studies, that would be great, which is good laboratory practice. Most of our labs does not actually have that level of documentation that we have a JLB labs. However, if you can focus on actually making sure that you are actually complying with the pharmacopoeial ICH guidelines when doing studies, for example, if you're doing stability studies, simple study like stability study, you want to go follow the ICH guidelines. That would have more value when the industry comes to review what you have, what you got to offer them, and they know that you know what you're talking about. And also, of course, you need to set up a deadlines and follow up with all your co-investigators. And once you accumulate certain research that you think you can build a claim around it, then you file a patent in a strategic way, because that's very dangerous, because your own patents later on, because as you develop, you file patents, but you want to make sure that they are very distinct, very different. Otherwise, the patent office, they can use your own research against you to kind of like, reject your patents application. This is a big story. We can elaborate a little bit about it later on. So if we're going to talk about patents, first of all, what is that I can patent? What is the requirements when it's a patent examiner is actually from the patent office looking at your invention? First of all, they want to look if it should meet certain criteria. Like it's first of all, it must be statutory, which means it's tangible, something eligible, something non-fictitious, right? It definitely has to be novel. It's a new, okay, and this is where usually they catch you with. So basically, should not be published before or disseminated before, should be useful, and also should be non-obvious, which means if you bring any scientist in your area, okay, can they come up with that invention without really going to extensive experimentation and creation of similar ideas or something like that. So basically, it should be non-obvious. Most of the time, we're going to talk about this rejection is due to lack of novelty and non-obviousness. And I've experienced a lot of this. I have like more than 80 patent applications. I've gone through a lot of examinations, and I mean, we filed many responses to the patent offices in order to argue about these 2 points. So what is that you need to be careful about when you are a scientist? First of all, you're not supposed to disseminate, publish anything that you want to patent. So you should not submit an abstract, publish abstract, you make a public presentation like this one, or any documentation that actually it becomes available to the public. Otherwise, you cannot patent the work. So the idea is that you need to file a patent, then publish your work. Yes, it's contradict the publication we all wants to publish, but if you want to go translational research, you cannot publish. So this is why you'll find people who are actually doing a lot of patents, they don't have a lot of publications. So it's until later on, because later on, once you file a patent, you can publish, and we're going to talk about this as well. So what are the different stages? Just to give you a brief idea about the filing. So first of all, you need to file a provisional patent. Usually, it's actually through the technology transfer office of your institution, or if you are independent, so you can file on your own. Okay, but then you have to hire a patent lawyer to file for you because it's really complex. The linguistic, the terminologies that they use is not something for a scientist. And the selection of the words is very important, especially when you're putting your claims into the patents. So basically, you can file provisional patent. Usually, this gives you a protection for like a year, or you have a 1 year span to add more information to it. It's not really expensive. It's around 5,000. The actual application is around 500, or exact is 450. I don't know if they changed it or not, but it's around 5,000. This is how much the attorney along with the application fee cost roughly. Okay, and then later on, after 1 year, you have the option either, or basically, if you want to continue filing the patent, you have to either file it in a specific country, or what most of the lawyer does is actually they file a BCT or a WIPO patent. So basically, a BCT application, it's an international patent, is going to give you protection for almost 155 countries, major countries, and these protections good for around 30 months, and that give you enough time to kind of like, market your invention, to try to decide what are you going to do about it. Are you going to start your own startup, or you want to license it to raise funds, for example? So it gives you a lot of time to kind of like, to think about how you're going to proceed with this patent application or the patent that you have. So then once you file the patent, you're going to get an office actions, many office actions. You get, first of all, the preliminary research reports. Basically, they're going to use this preliminary to kind of like, to try to reject the patent, to say that it's not novel, or it's not, or it's obvious. So basically, you want to prepare yourself. There's different types of office actions that you can get. There's the first one, the second one, and then you might get the third final rejection letter. Or initially, usually what they do, they have a letter of restriction. It depends on the strategy of your patent lawyer. If they're combining different aspects, different claims into one patent, then they're gonna split them into multiple patents for the same project, because sometimes one aspect talks about composition, the other aspect talks about use, the other aspect talks about application. So they ask you to split them because you cannot combine them. And this is what creates a patent family for the same project. Then you get either multiple of actions, usually two, and then the last one would be a final rejection, or you will get a letter of allowance, which is usually within 6 months. You can, if there's no objection from the public, you can get granted. The process of patent filing might take from 2 to 7 years. I got one patent took like almost 7 years to get issued. The other one, others took like on average 3 years. This is the average time. So basically, it takes a really long time before they get issued, if they get issued. Usually, as I mentioned, they attack you through the lack of novelty and the obviousness part, where they want to, the patent officer's job is to show that yours is not novel enough, and you want to convince them with evidence that is novel. Same thing about the obviousness. Who owns the patent? So at the end, if you are in the institution, the patent might not be yours. So we have in the patent, the first page of any patent, you'll find there's the inventors, who are you and your co-inventors, the applicants, the people who applied for you. Most of the time is the institution, unless you are applying for yourself, which has cost a lot of money later on. So it's not really advisable, because this is a high risk type of business. You have the patent attorney, the information of the people or the attorney that actually office that actually dealing with your patent, and the assignment. The assignment is actually who owns the patent. So if you see, for example, University of Sharjah, that's mean the patent is owned by it's owned by the university. Now, most of the institution, they have like some sort of like bylaws to divide or share profits of these patents. So again, this is something that this is an area of your interest that you want to explore before getting employed in one of these institutions, to see how they deal with patents. If you are individual, like independent, then the patent is yours, so it's under your name. But most of the situation, it's not the case. It's usually with other institution, or assigned to a private company. If you're an employee of a company, most of the time it's 100% for the company, so you own nothing. So are you a co-inventor? This is a very tricky question. And if we have research assistants with us, or postdocs, maybe this is an area that they need to look at or consider. So when you are a co-inventor, you are a co-inventor when you contribute into the intellectual part of the invention. So conceptualization of the idea is very important, have the heaviest weight, okay? Conceptualization of the method, the approach, the study design, how it's being performed, okay, is very important as well. Data analysis and how they can be, what is the interpretation of the data and the understanding of the data, how you can actually come up to conclusion using these data. This is a lot of intellectual contribution. However, if you are only contributing to the work, actual work, like executing the experiment, collecting samples, analyzing samples, spending from 8 o'clock in the morning, 8 p.m. in the lab, doing analysis, standardized analysis, not the ones that sophisticate one, then you are not a co-inventor. So basically, it's mostly about the intellectual contribution, not the actual physical work that you've done in the lab. Okay, unless you change something that require intellectual contribution, that actually change the design or methodology and so on. So that was actually the patents. But who is doing this? Usually an institution, the technology transfer office. They serve many purposes. One of them is actually to file for your patent. Secondly, they can market for your patent and try to communicate with interested parties. They can do a lot of commercialization activities in addition to the assessment. Also, they can work into or help you with the negotiation. Like all my patents, the licensed patents are licensed, is actually being negotiated by the Technology Transfer Office, because I'm not an expert in contracts. and terms and agreements. So basically, you need a lot of help. There's lawyers, there's people who are actually, they've been doing this for many years, they have a lot of expertise, so they can help you. And also, they work, the Technology Transfer Office would work to maintain the relationship with these companies and follow up to get updates. And also, they can guide you and support you. Like, do you start a startup? Do you actually know license it to a third party, to a pharma company, for example? So they can actually guide you in this perspective, and also it has to do with how they see the future for this product. So they have a lot of expertise. You have to build a really good relationship with the Technology Transfer Office people if you are planning to do translational research. So do I license it, or do I start my startup, my company? Again, institution varies in their flexibility in allowing you to start a startup. Sometimes you have to leave your job in order to start a startup, okay? If you have the patent rights for you, if it's being released to you. Sometimes institution can release it to you, okay? Or sometimes they keep it. If they keep it, you cannot do startup unless you are in agreement with them. But do I do licensing or startup? What is the difference? Licensing, basically, that means that you are having a contract with a third party, with a company, with a pharma who has the expertise to do product development. So basically, you can have a contract for a period of time for a specific application or field, and for a specific region or worldwide. Again, this is a contract, so you can design it the way that you think that fits your interest. But then in this case, that's it. You lost ownership of the IP. Whether you or the university now belongs to that company. But what is the advantage? The advantage is that you can reach to the market quickly, right? So they have the expertise, they have the infrastructure to develop the product, they have the channels, the marketing team, so on. Financially, you get kind of like, passive income. So you can get it from licensing fees, from annual renewal fee, from milestone payments, again, based on the contract terms that you developed, right? And based on performance requirements that you actually designated in the contract. If it makes it to the market, you can get royalties. And royalties divided between all the patent, kind of like, co-inventors and the institution, if you're working in an institution as well. So usually royalty can vary between 2% to 10% of the sales, which is not bad at all if you are an individual. Actually, I was hoping that one of my products would make it to the market so I can retire, early and maybe just do only research. But I mean, still, I'm not that lucky enough. Anyway, so basically, you have low risk. You don't have any risks. You are actually just gave the job to somebody else who have a lot of expertise, hopefully that can move it to the market. Your involvement and the effort is very low, mainly maybe in the first year. All the patents that I licensed, I was engaged in the first year when we're doing the technology transfer, and they understand every part of the product development, and then they put their own knowledge on top of it to make it to the next level. Control, you have no control in the decision making. They're making all the decisions. However, on the other hand, if you're creating a startup, then you are and you are actually can start your own company and you own it 100%. Now, the problem is that financially, you need to be financed, you need to raise money, maybe multiple cycles, but you're kind of like building growth to that company. And at the end, if there's any profit gains, it's actually 100% for the company that's owned by you and your co-inventors. But the process is very slow. You kind of like you need to build the knowledge to how to develop the product. You need to hire people, to start the company. You need to have enough sufficient funds to pay them for salaries and so on. Risk is extremely high. So chances to make it to the market is very low. Like, I mean, startup success rate is 10%. 90% of the startups fails. And usually majority of them, they fail after 2 years. They call it the death valley. The first 2 years, the death valley. This is where most companies, they die after 2 years. The effort is huge amount of effort because you're building a whole company with the people, with the development of the product, with all the steps that are required by regulatories. But at the same time, you have full control of the product development. So what is that you're going to do next? Whether you decided you're going to go with the licensing or a startup pathway is the same for product development. You have to go and file. So basically, when you're filing an IND, investigation new drug, and this is basically the part that you can see here, the clinical trials, the ones in the middle in the graph, to get permission from the regulatory agency to initiate clinical studies. You cannot start clinical studies until you actually provide them with information that they would understand what you're doing and give you permission to make sure at that stage they're trying to make sure that you're not posing any high risk on the human subjects. So basically the risk is reasonable and controllable, and you have a risk management plan and so forth, right? But also you need to build for that. In order to approve you or to give you permission, you have to give them data. And this is where the first two kind of like, or three boxes that you see in the graph or the chart, basically to build the basic science. So you need to have the science behind and the information and the animal studies to support the approval of that clinical trials. So if you are actually, when you're doing basic research, you're actually doing the studies to serve that purpose, you save a lot of time for the company that's going to take it to the next stage, or to the startup that is going to take it to the next step. So again, this is why it's important that you purposely design the study to serve the IND filing. So here you can, what is that? Why we need the IND filing? We need the IND filing for, as we said, to initiate clinical study, but for what? For any new chemical entity. Okay, any new drug. And usually this is the longest and the most expensive pathway to have a new chemical entity, because you have to prove their safety and so on. Or you are actually going to try it for an indication that was not approved from before. So it's a new indication or a new dosage form. It was an injection, now it's a tablet. It's a new administration route. For example, it used to be a parenteral, now it's going to be oral. Dosage level, so is the strength that you're going to use it, and so forth. Or it's a combination product. So all of this require IND filing in order to initiate the clinical studies. Usually this is the IND form. There's a lot of information here. I'm not going to go through it, but if you go onto the right side, you'll see content, which is number 14. This is all the information that you need to provide along with the application form. So this application form, just to tell the regulatory agency, the FDA, for example, what is that you're applying for. The second page, it tells you what is that you need to do. You see from 1 all the way to 12 items, or basically titles, big titles that you need to include. The part that I want to highlight is number, of course, you have 6, which is the protocol about the clinical trials that they're going to allow you or give you permission to proceed with. But also they're going to ask you for number 7, which is the CMC, or the chemistry, manufacturing control. And this has to do with your formulation. So again, you save a lot of time when you're IND filing if you have this information under the basic research that you have completed. Also, pharmacology, toxicology, you can have good amount of that information, basic information that can serve these chapters of the application. So again, you're building value to the product, you're expediting the process, you're providing the company that's going to license it or the startup some basic information that they can rely on their application. Phases, usually drug discovery takes from 3 to 6 years. Then the clinical trials, it depends on the condition at the term of the trial. You have phase 1, 2, and 3, different population. And then you have the approval, usually takes up to 2 years. But then you can do the post-marketing, or basically the phase 4. What is the chances? Now, all this basic research, for example, I put here percentages. Research, for example, which is the drug discovery part, which is the pharmacology, medicinal chemistry, very low chance to make it to the market, because you did not even try it on animals. You did not characterize, you didn't put in the formulation. So you got 0.001% of going to the market. Pre-IND, which is the formulation part, the drug delivery part into animal, then you have 1%. Once you hit phase one, once you go to clinical trial, then the percentage goes significantly as you proceed from one step or one phase two to the another. And of course, it builds value as well. So as you go, because your success rate increases, the increase, if you want to license it, increases the price or the value of the product if you want to license it to another company. So what are these clinical trial phases? Maybe you're already familiar with most of those. We have phase one. The idea is to prove safety and basically smaller population, 20 to 100 healthy volunteers. These are not patients, these are volunteers, so they don't have the disease. And you start the dose from a very low dose, and then you start increasing. You start from 1/10 of the highest no effect dose in animals. Again, if you didn't do the animals, that takes longer time, so you have to do the animals and do dose response or dose effect or dose escalating studies. And in this case, you see where is the highest one that did not produce an effect. This is the 1/10 of it of the dose. You start with the human, and then you start increasing the dose until you see some effect, some pharmacology. What is that we're doing these studies? We're building safety, but at the same time, we are learning about pharmacokinetics, about absorption, distribution, metabolism, elimination, pharmacology, structure, action, relationship, and so on. So we're building a lot of knowledge from phase one. Then we move to phase two after we build certain confidence about the safety, but now we move to patients. So we're gonna try to apply, and this is where we talk about translational research. We move into clinical trial, okay, and then you put it into, give it to patients, usually several hundreds up to 500s or a little bit more patients, and they are divided into phase 2A and 2B. 2A is about dose response study. So basically here you're trying to determine the minimum effective dose, right? So now, but this is on patients. So you're going to see the efficacy. You want to see if it's actually curing the disease, right? But what is the minimum effective dose? Because you started so low and there's no effect, no effect, no effect until you start seeing an effect. And at the same time, you're seeing building the profile for the side effects and the safety of the product as well. Then you move to phase 2B, and this is very critical because this is where you're going to develop the therapeutic index, because you're going to do a dose ranging study. You're going to start increasing the dose to see the effects and correlate the dose to the effect to the side effects. So kind of like, building now, what are my therapeutic index or the strength that I'm going to use and apply in phase 3, and you start and keep increasing until you start seeing some kind of like toxic effects that you don't want or some side effects that cannot be tolerated. Then you move to phase 3 again. Phase 3 is a 3A and 3B, and here you already build safety profile, you already build efficacy profile, but on a smaller population. Now we're going to try it on a larger population, could be thousands, in this case up to 1,000 patients. And again, what you're trying to achieve here is basically to establish the benefit to the risk relationship. We're not trying to establish efficacy. Efficacy already being seen in phase 2. We're not establish safety. Safety being seen in phase 1 and 2. Right now, we're trying to justify and say, you know what? These side effects that we see, we can tolerate for the amount of benefit that we're gaining from that product. And then you can move on to 3B. When you move on to 3B, you can actually start filing your new drug application. This is to seek approval from the regulatory agency. I said, I did not complete the studies. Yes, but this type of study, 3B, is not intended to prove efficacy and safety. We already established that. It's intended to kind of like give more information that required for the approval. So you're not going to get approved until you finish phase 3B. But at the same time, you can start the application process. It takes a long time, up to 2 years, so you can start the process. Now, this study can help you with getting more information that would help with the prescribing information. So all the label information can be generated. Some of it generated. Are you going to take it with food or with no food? What's going to happen if you have a liver function? What if you have, for example, poor kidney function? So because you need to adjust the dose. What about the quality of life? And also some other if you want to explore some other indications. So the results from phase 3B is not going to change the decision about if you're going to approve it or not, but it's going to provide information that we're going to need in order to put into the market so the practitioner would have more information. Once it's been approved in the market, we can do phase 4. Okay, and this is again to gain more understanding, to try to apply it to different kind of like, try to explore certain side effects. And of course, there's a reporting process. I don't want to go through too many details here, but there's a reporting process that you need to have an open communication with the regulatory agencies during all the phases, from one all the way to even after being in the market. What other requirements? So it's how I'm going to conduct the clinical trials. I'm not going to take a product that actually I did a preclinical formulation in my lab and give it to human. It should meet certain expectation. So first of all, when I'm going to do the clinical trials, I'm not giving only 1 or 2 patients. I'm doing thousands of patients. So I need to scale up studies in order to kind of like, manufacture larger batches, make sure that still have the same criteria and controls that we decided from before. Also, I have to follow GMP manufacturing guidelines. GMP, it's a good manufacturing practice guidelines. It's required for the manufacturing and for the facility. So basically here, any product that's going to give into human, it should actually manufacture according to GMP. This will ensure the quality of the product from an ethical perspective, manufacturing perspective, from control perspective, from stability perspective. So all of these needs to be ensured, whether you're doing phase 1 or 2 or 3, or even you're giving products that actually been already approved into the market. Now, if you're doing blinded trials, then I'm gonna definitely do some comparison. I have my investigator drug, the new drug I want to get approved. Maybe I'm using the placebo, I'm using references, but I'm gonna do blinded trials. So all the three, they need to look alike and taste alike. So basically, patients cannot differentiate, right? And in this case, again, manufacturing a third-party facility would be very useful because they need to custom design these products to suit these trials. It's something they cannot do in the lab. You have to have specialized large-scale pharma companies that they are doing this business for years. Now, after you're doing that, you have concluded all your clinical trials, you gained all the information, you establish what you need to establish, then you're ready to file for the NDA, new drug application, or the CTD as well. I'll explain in a minute. So here, the whole objective is that you want to show to the agency that's going to approve your drug that your drug is safe and effective, right? And the benefits of that product outweighs the risks. Okay, and also you want to provide sufficient information or data that actually matches the information that you include in the labeling. So the label or the package insert is actually have a sufficient information, required information that actually comply with the results, or basically as a result of the results that you actually obtain from these clinical trial, and they are appropriate. Also, they want to make sure that when you're manufacturing the product, because they're going to go and visit the site, okay, so that the manufacturing of the drug and the way you're going to control for it is actually create a high quality product. So all the strength, the identity, the quality, the purity is actually being preserved during the manufacturing. So this is the NDA form. And again, I don't want to go through the details. Usually we spend lectures and lectures. We actually deliver I deliver a regulatory course, part of the master program that we offer in the College of Pharmacy. And we talk about all these different chapters that we need to include, or sections that we need to include in the NDA application. Usually there are 20 sections if you are filing to the FDA, but also there's something called, maybe you heard about, which is the Common Technical Document, or the Electronic Common Technical Document, because now it's filed electronically. This is being developed to kind of like, to harmonize and streamline the submission. Because if you're a pharma and you want to get the drug approved in USA, then in Europe, then in Japan, each one, they have their process and way of application, and this is intensive amount of work and data that needs to compiled and submitted to the agency. So repeating the work every time we register in a different continent, that or region, it's very challenging. So they came up under ICH, which is the International Conference of Harmonization, to streamline the submission process, and they're now following the Common Technical Document, the electronic filing for that. So in conclusion, or basically before we conclude, this is the common technical document, slightly different. The NDA 20 section, these are 5 modules. Again, they include the quality, which is the manufacturing and control and the chemistry. They include the non-clinical information. They include the clinical information and so forth. Again, I don't want to spend too much time on this because, again, we spend lectures to talk about the different sections and how can we organize the information to be reviewed by the reviewers in the agency, like the FDA or EMA. So in conclusion, as you've seen, the translational drug development research is actually very rewarding, very impactful, can actually affect the patient health directly. It builds a really great reputation for the scientists involved, for the institution where they work. You become very well known, very popular, right? And you go into the news and so forth. And also, it's impact the economy. It boosts the economy, creates a lot of jobs. So basically, it has a direct impact on the region. But at the same time, it's very challenging. It's a high-risk business. Failure rates are very high. You spend millions of dollars, and then you might not end up with any profit. So this is why sometimes the prices for these products are high. But at the end, it's that kind of research it's a translational research was purposely designed. So you should carry on the research with having in mind that should meet the certain level of expectation by the pharma who's going to license it, or is going to serve you later on if you're starting a startup to kind of save some time, regulatory time, or basically to meet the regulatory expectations. Honestly, the institutional infrastructure, the environment, the level of understanding should be there. They should be supportive. Otherwise, it's very difficult to carry that kind of work because it's focused, it's designed purposely. Sometimes you're doing studies for the purpose of translational research, but if you want to publish it, they will not get published because it's just not enough, but you need it to serve certain purpose. So again, you need a lot of support from the surrounding environment. I'm sorry if I took too much time, but again, I would like to thank you all for your active listening, for being here with us tonight on a Thursday night. And if there's any questions, any recommendation, any suggestions, please, the floor is for you. I think Professor Bashir can moderate
Yeah.
any questions. Thank you very much.
Yeah, sure. Thank you very much, Prof. Mark, for a very comprehensive, informative presentation. You took us through very different areas, but it's much needed. I was so interested in the first part when you were talking about how it's very important to build this network with practitioners and with clinicians in the early stage of the project. And I think this is very important. And, like one of the aims of CREATE is about, kind of bridge this gap. Now, most of the time, I think scientists, after they prepare the protocol and after they finish the whole thing, and if they want to take it to, to do some recruitment or to, discuss some patients, then they start thinking about a practitioner or about the clinicians. But what you said about when the clinical teams involved in the initial discussion of the idea, that will enrich the whole process because they
Yeah.
do have gaps themselves in their practice. So it's ideal, but how to facilitate it, how to kind of convince more people to do that.
It's a great question. It's actually, as I mentioned, it's you can the whole project can fail if you don't start right. if you don't have the team, the proper team, the people who actually deals with the issues on a daily basis, as I mentioned. I mean, you might spend years working on a research, but then in practice, you find it that it's not really useful. It's this is
Mm,
not the way they are doing things in the clinic, right? So basically, you need to have like, close relationships. You need to target. You need to go to look at in your area who are like the practitioners that are interested in research, who are like they have high volume of patients. Sometimes they might not be interested in research and they might not want to be part of it. They don't have time for it, but it's fine because you're just going to consult with them. This is why I said you can't have them as a consultant, right? Don't go and assume that this is the way it's going to be done. Okay, you need to go and ask them and try to have a chat with them. Sometimes if you build some network of friends that are practitioners. For I'm going to give you an example. Like when I was in the States, in Florida, I have like a group of friends, and happens to be all of them, they are practitioners in different fields. So when we sit to have coffee, I kind of, they don't know what I'm doing, but actually I'm trying to get some information from them to ask them, okay, what is the things that you have a problem in the clinic? Like when you're dealing
mhm,
this, they start talking about the appointments. I said, no, no, what about the medications? What about the application, and then this is when you probe them. This is when they start, and I explain why I'm doing this. One of the first idea, which is when I was working on anaphylaxis and trying to come with a new, a novel, route of administration and dosage form, it was the idea came from a physician. It wasn't from a formulation person. But at the
mhm,
same time, I see the other side. I have, for example, I'm collaborating with other people who are physicians, and they bring an idea. It's a nice idea, but you cannot do it. In the lab, you
mhm,
cannot experiment it. So you need both. You need both ways. You need the formulator or the scientist in the lab who knows how to experiment things, okay, and the ideas from the practitioners who sees the problem. And preferably, they should be a team. There should be a team. You should not be working in silo and by yourself and you think you are the most knowledgeable person. Nobody knows everything. It's about collaboration. So that kind of
yeah.
environment would enrich the whole project.
Yeah, sure. I think we got a question from a student because he's asking that can a student work on a pattern alongside with a supervisor? Yes or no, and how? If the answer is yes.
I'm sorry, can you repeat that? A student
Yes,
working with.
we got a question. Yeah, we got a question, I think, from a student because he
Mm.
asking or she's asking, can a student work on a pattern along with the supervisor? Yes or no? And if yes, how is possible?
So honestly, to put it straightforward, most the time, but there's exception, but most the time, it's very difficult to include a student, especially like a master or PhD students, as a co-inventor, because at this
Mm-hmm.
stage they might not have a clear idea about the project, or they come up there. The idea usually comes from the supervisor, but that's not necessarily all the time the case. Like for example, my first group, my patents, I was a PhD student, but it was my idea. So I
Mm-hmm.
proposed the idea to the supervisor. I told him, let's try this. And he was like, not really very confident. I was like, no, no, let's try this. I can do. So basically, you don't want to be, if you are a student, you don't want to be a passive student. You want to be an active student. You want to be reading, educating yourself, proposing idea, even if it's the main idea from the supervisor, right? You can
Mm-hmm.
go and take this idea and search it and look what others are doing and come up with proposals and come up with
Mm-hmm.
alternative ways, come up with ideas to do. Many times PhD students are capable of coming up with testing methods that we did not think about, or basically we are used to do things certain way. They come and they say, oh, we read this article and this article, and they're doing this, they're trying this, how about we try it out? Now you are a co-inventor. Now you are a
Mm-hmm.
co-inventor. So being an active student rather than passive student, that what makes you a co-inventor. It's possible, and your
Mm-hmm.
name can be there in the patents. I put two of my students. In some of my patents, two of them, actually two group patents, they already been issued, and these were they were my PhD students as well with me as a co-inventor. So it's possible.
Okay. Thank you very much for that. And I think now we have a comment actually from a clinician saying that this is a very sensational scientific talk, which he recommends every clinician like himself to attend such a presentation. So thank you
Thank you very
for
much.
that. Yeah, thank you for that comment. And it was sensational actually. Another, like for example, like it's a general kind of question because most of the time scientists work in studies to publish it. So publication seems that to be the common outcome for most of the projects. But from your experience, when do you think, what you recommend for the scientists, when they should think about filing and whole process, when they think that this idea or this project is not for publication or for
Yeah.
something else? I know it is not just one factor will lead to that, but from your experience, when you can tell or when you can
Yeah.
think this is something is very special.
So first of all, like I mean, I guess because I have now good experience with patent filing, so I have this level of understanding, and I can tell from the beginning this is a project
Mm,
for a patent filing, or this is a project just only for publication. And I know I kind of like have six numbers of projects, and I know this is going to be for patent filings. And for patent filings, you don't need a regular students. You need a students focus who can do things really quickly because this is might impact their publication ability, right? So I
mm,
know, for example, I save these projects until I see the right student, and then I say, how about this project? And then I
mm,
align that project with that student because with the problem, and this is I suffered from, but there's no way around it, is that when you're filing a patent, you're delaying publications. And when you're
mm,
delaying publications, it's a catch. I mean, you have a student who wants to graduate. So you cannot,
mm,
independently, like we cannot just delay it forever, right?
mm,
You need to reach a stage, or you find alternatives. For example, find
mm,
another publication. For example, University of Sharjah, they have to publish. So you can publish something else, okay, with the students, other project, a side project, something else, and save that one for until you're ready to file a patent. Because filing a patent, you cannot just file anything because you can
mm,
damage the future of the patent later on if you just file very quickly and then you need to file again, and then that second filing would not be you might get the first one approved but the second one rejected, and the whole project is not complete. So you had my approach is that to file complete story as much as possible because I suffer a lot
mm,
from being the patent officers like using my own patents against me, and now I try
mm,
to explain, no, this is different, this is not the same,
mm,
it's not like
mm.
published before. So it's
Yeah.
basically you're limiting sometimes your ability to publish. And again, this is another thing affected me. I was like sometimes like earlier in my career, I was like, how come you don't have a lot of publications? I said, yes, I don't have a lot of publications, but I have a lot of patents. I was like, but your
Mm.
H index is very low. I was like, but patents does not go into H index. So sometimes you suffer something.
Yes. Yeah, I know it's very difficult to balance between the two,
Yeah, but at
especially
the same,
for
yeah, but
the
on the other
amount of
hand, I have a
effort.
lot of patents
Yeah.
and I have license, so I have, generated some income. So basically, always you cannot get everything in life.
You cannot get everything, definitely. And I just for the sake of time, we have one last question and one comment, and then we'll close the session. When you talked about the confidentiality, and it's very important when you submit your idea, especially if it's very kind of special idea,
Yeah.
you have to make sure that the institute has some sort of agreement with the reviewers. Is
Yes.
this something that's common practice? Is something that you can find
Yeah.
out, or it is not? It is something so ideal to
That's
know if
really
the reviewers who look after your projects are having an agreement,
that's that
confidentiality.
that's an amazing question, and we've been caught through this before. So if you're doing this kind of research, the translational, you cannot really apply for grants. unless
Mm.
that the funding agency or organization have a confidentiality agreement with the reviewers. So when before they're sending the grants to the reviewers, okay, they make them sign confidential agreements. So basically, they cannot use the ideas or any of the proposed information in the proposals, okay, on their own. So
Mm.
most of the like in when I was in the US and Canada, most of the institution they have that. I have only one
Yeah.
situation where basically they didn't have or we didn't get a clear answer. And in this case, we had to rush and file a patent overnight before we submit for the grant because to meet the deadline. Same thing here in, for example, in UAE, I believe you need to find out. I think some of them they do have that. So
Mm.
basically, it's very important if you're thinking that this is a translational type of research, find out. Find out if
Mm.
they ask the reviewers to sign such an agreement or not. Otherwise, you're disclosing your novel ideas or what you think is going to be a novel idea to the public. And that researcher, and also I suffer from this, that researcher have used your idea. I have a situation where actually I discussed something with people that I felt like, I can't trust, and they took the idea and they moved faster than me and they filed the patent. So it happens, when you are in this field
Mm.
for a very long time, things
Mm.
happens.
Yes. Yeah, and there is, like people asking about the ethics and, the ethical issues related to that. Obviously, like when we start the whole kind of foundation for our webinars, we had two webinars about ethics and foundation of ethics. So
Yeah.
I just referred these people to the EDAS YouTube channel to see all this. And also, what we have in this set of webinars, we will be having a speaker will talk about the local regulation when it comes to ethics in research. So please just, stay tuned with us. I think we will close the session here. Thank you very much, Prof. Mark.
Thank
Again, we
you.
really enjoyed your talk. And thank you very much for investing your Thursday night with us. Wishing you all the best. And definitely, we will be happy to have you once again in the future. Thank you
Thank you.
very
Thank you
much.
very much. I really appreciate the invitation and thank you very for the attendees and have a good night.
Yeah, you too. So just to give you an idea about what we're going to do next, we do have still more kind of upcoming webinars. We will be talking about the power of cohort studies. Again, a very selected topic for the people who are interested on that. After that, we will be talking about cross-sectional studies. And then sometimes in June, we will be talking about how to bring participants on board when we talk about the recruitment of patients. And as I just mentioned, we will be talking about the research ethics in DHA, which is related to the local kind of guidelines. Our next webinar will be on the 30th of April. Prof. Farah will be talking to us about time, exposure, and outcomes, the power of cohort studies. Thank you very much for everyone. Have a great evening. I'll see you next time in our upcoming webinar. Thank you.
Thank you.